B-Day. So we're going to resume our wander through chapter 15. And in this part, we're going to continue to talk about the molecular and cellular details of how different pathogens make us sick. So one of the things that can happen is that a bacteria or a virus or some other pathogen causes actual damage to our cells. And this can happen in a number of ways, either by consuming nutrients that we need, so we sort of get a kind of malnutrition. Pathogens can directly damage the tissue in the area where they are infecting your body. And systemically, if they produce toxins, some of these toxins can get into your bloodstream or your lymph system and travel to distant sites. So if you get a wound and your pathogen makes a toxin, that even though the actual pathogen itself is local, the toxin can travel through your whole body and have wide-ranging effects. So let's first talk about consumption of host nutrients. The biggest problem usually is that bacterial pathogens are stealing your iron. So bacteria need iron to grow just like we do, and they need it for a number of processes, most importantly for respiration, if they do that. So if they don't have enough iron, they can't grow properly. So they're really good at stealing it from us. They secrete something called siderophores, and you do have to know what this means. It's just a type of chemical that's really good at scavenging iron. And so what they do is they secrete the siderophores, and they steal your iron from the blood. And some of these pathogens will first explode the red blood cells in order to get that iron. Okay, so we know that that's the case. We know some pathogens, human pathogens, that will explode red blood cells, and a process that we've seen on blood agar plates, right? If we're talking about the direct damage they can do to tissues, then typically this is a result of the pathogen releasing waste products. And also if it's a virus or some bacteria, some of those will also explode the cells, so rupture the cells. And where the infection is active, the cells are directly damaged. There are also toxins that can be made. So here's just some definitions you need to know. A toxin is defined as a substance that contributes to pathogenicity. That's sort of a circular definition. Toxogenicity is the ability to make a toxin. Toxemia is if you find this toxin in the blood of the host. What is a toxoid? And we'll talk about this again. It's basically if you take the toxin and you do something to inactivate it. And typically you're doing this, why? Why would you do that? Yeah, to make a vaccine, right? So you take the thing that would normally make you sick, we inactivate it. We'll talk a little bit more about how you would do that, but does anyone have like an idea right now of how you might accomplish that inactivation? Say again? That's okay. So a lot of times a brief heating, right? So it's one way to think about that is when you go to heat fix your slides, you're sort of preserving the shape of the cells without giving it so much heat that you incinerate it. And so that is sort of analogous to how you would use toxins. You would inactivate them, sort of make them, damage them a little bit so they can't work, but so they still look like what they look like. And then that means that when your body sees it, they can make antibodies against that thing. When you do that process, you inactivate your toxin, making it into a toxoid and making it into a vaccine, then the antibodies your body makes are an antitoxin. Okay, so we have to talk a little bit more specifically about toxins. As I said, they can be transported systemically through the body, even if your pathogen is localized. If they're making toxin, it can travel through the body. And the result can be a number of things, but it can include things like fever, diarrhea, shock, heart and nervous system problems. There are about just over 200 types of toxins that are known, and just under half of them have their effects by damaging the cell membranes. There are two general types, exo and endo. Exotoxins and endotoxins. So what is the difference? Outside and inside, okay? Now, more specifically, when we say an exotoxin is outside, what does that mean? Where was it made? Where is it going? Right, it's made inside the pathogen and then it gets sent outside, exported. Now, what about an endotoxin? Yeah, that is a component of the cell that's actually toxic itself. So what the implication is if you have a pathogen that's living, it's able to make exotoxin, then that's when it's most dangerous. But if it's an endotoxin that's causing problems, the most dangerous time is when that pathogen has started to die and the endotoxin is being released. So this is just a schematic to show you that difference, that exotoxin is being made by a living pathogen and secreted outside. The endotoxin is this piece of the pathogen which is normally inert. It's just sitting here doing not a whole lot, but if this cell were to die and that piece is released, it starts to cause problems. And all endotoxins are found in gram-negatives, and they're part of the outer membrane. So remember the structure of the gram-negative, right? We've got, just like all other prokaryotes, we have our cytoplasm, we have our plasma membrane, right? Then we have a cell wall, and the cell wall has this very particular structure. It has a little bit of peptidoglycan, but not very much. And then outside of that, what is there? This extra lipid bilayer that looks a lot like the plasma membrane, this extra protection. It's almost like the envelope of a virus, but different, obviously. And in that outer lipid bilayer, there is something called lipid A, and it's in the lipopolysaccharide layer, that outer membrane of gram-negative cells. So first, let's talk about exotoxins. Their action is specific, okay? What they do to the host is specific, and there's a number of different options. Now, the way that we name exotoxins can vary depending on a number of things, but sometimes they're named by the type of cell they affect. So you may have heard of a neurotoxin, a cardiotoxin, a hepatotoxin, leukotoxin, enterotoxins, and cytotoxins. So what do these do? What is a neurotoxin targeting? Neurons, okay? Cardiotoxins. Cardiotoxins are infecting what cell type, do you think? Cardiovascular, your heart cells, right? What about hepatotoxin? Liver, yes. Leukotoxin, anybody know what that is? White blood cells, yes. So these toxins will kill those cells or affect those cells. Enterotoxin, that would be your gut, okay? Like the enteroviruses are gut viruses. And cytotoxin, that is basically saying it's killing cells. It's a generic term. So some toxins are named instead by the disease that's associated. So we have diphtheria toxin and tetanus toxin. What are the diseases associated with those toxins? Diphtheria and tetanus, right? It's not a hard question. Other times we'll name the exotoxin by the bacteria that makes them. Okay, so botulinum toxin is made by what? Clostridium botulinum. Vibrio enterotoxin. If you've heard of Vibrio cholerae, that is the pathogen that causes cholera. So what are these exotoxins? They're always proteins, which means what? Where did the instructions for making those proteins come from? They're encoded in the genome of the pathogen. Now, the thing about exotoxins is because they're secreted, and because of the way that they work, even very small amounts can be harmful or fatal. So, for example, one of the most dangerous toxins is botulinum exotoxin. 1 milligram of that will kill 1 million guinea pigs, which is a weird statistic, by the way, but it is at least something that you can sort of imagine. Now, the gene for this exotoxin is usually found on a plasmid, or it's transmitted between bacteria via phage infection, right? That process of transduction that we've talked about. Once it's made by the bacteria, it's soluble in the lymph and the bloodstream, so it gets all over the organism. And there are 3 ways that different exotoxins work. So I'm not suggesting that every exotoxin does all 3 things. So some of them do one, or some of them do the other, and some of them do the third thing. So what are the modes of action? Some of them inhibit metabolism, usually by targeting, say, the ribosome and shutting down protein synthesis. So if you're a host cell and you encounter a toxin that shuts down the ribosome, what will happen to you? very bad things, right? Because our cells are constantly making proteins. That's how they stay alive and how they adapt to their environment. So some exotoxins instead just destroy parts of the host cell, and others of them have a more systemic effect where they overactivate the immune system. We'll talk about all three of these. These modes of action coincide with the type of exotoxin, and you'll see that in a second, but there are three types of exotoxin. There's the AB type, there's membrane disrupting exotoxins, and then superantigens. So you will have to know all of these and how they work. We're going to talk about that now. The most difficult one to remember is the AB exotoxin. And so here's a schematic of how it works, and I'm going to walk you through it. First of all, the AB exotoxin, as you might imagine, has actually two proteins, A and B, that are bound together. And the B part is the part that's the binding part. So on top we see the bacterium, and it's making this AB exotoxin, sending it outside, and the B part is what binds the cellular receptor of the host. Okay? And then that triggers the host to take up the toxin. Once it's inside the cell, then the two parts of the toxin separate. And the A part is now free to do what it does, which is usually the A part is an enzyme that inhibits protein synthesis by perhaps binding to or chopping a piece off of the ribosome. Here are some examples of AB exotoxins. So diphtheria, botulinum, tetanus, and Vibrio, the toxin that's involved in cholera. All of these diseases make these toxins, and they have all acting by this effect. So it's a pretty potent method. The other kind of toxin, exotoxin, is membrane disrupting. What do you think that those do? What component of the host cell are they affecting? The membrane, okay? So when we say membrane disrupting, this is actually kind of a soft way of saying what actually happens. It's as if you took a machine gun and blew holes in your host. So what they do is usually they make proteins channels. So the toxin is a protein, and it gets into the membrane and it forms a pore, and now it's as if there are holes all over the cell. So here are some example leukocidins, hemolysins, and streptolysins. This is involved in streptococcus species that cause beta hemolysis, like we saw in those blood agar plates. Why is this a problem? If you blast holes in the membrane of a cell, why is that going to cause problems? Yeah, stuff's going to leak out, and other stuff's going to leak in. It's a really bad situation. So that's what some of them do. Now there's others that disrupt the phospholipid bilayer by chopping off the head groups of the phospholipids, chopping off those hydrophilic phosphate parts. And one example that's really bad is the organism that causes gas gangrene, which is Clostridium perfringens. This is an illness that was seen a lot in warfare when people were, in World War I and World War II, people were working in trenches a lot, and that exposes you to the soil. So you have a wound, but you're in the dirt, and then you get this in your body, and this is what happens to you. So this gas gangrene, the toxin that it makes chops the head groups off of the phospholipid, and entire massive parts of your tissues just fail, basically. It's really pretty nasty. Now the third type of toxin is the superantigen, which acts like a superantigen. So antigens are things that stimulate our immune system to respond. Particularly what they usually do is they stimulate our immune system to make antibodies. Superantigens will cause us to have a massive immune response. And if you think about it, why is it bad to have, why do we have allergies? We're having inappropriate immune responses to something that sort of is detected as foreign, and it's pollen or whatever. And so we have this immune response. So this is like having the 100,000 times worst allergic reaction that you can imagine. So it invokes this really intense immune response, usually by causing the T cells to over-proliferate and produce way too many cytokines. So what are cytokines? They're just chemicals that T cells that are activated produced to say, hey, there's a problem. Let's do something about it. And it causes all of these symptoms: fever, nausea, vomiting, diarrhea, shock, and sometimes, in extreme cases, death. Now, these are all things that you've probably experienced, except for the last part. Probably haven't had that one yet. But if you've ever had the flu or something, so your immune system, when it's battling certain pathogens, will have some of these symptoms, some of which are the result, actually, of your immune system. So it's like the pathogen makes this toxin that causes your immune system to overreact. So an example is Staphylococcus food poisoning, or Staphylococcus species that cause toxic shock syndrome. Both of them are, the main problem is a toxin that happens to be a superantigen that's made, and it's an exotoxin. So a couple of things: they're disease specific, and the effect of the exotoxin is not due to the bacteria, the live bacteria that's in your body, wherever it is. It's not that the bacterial cells directly themselves are causing the main problem, it's the exotoxin that they secrete. When we have a food poisoning situation, for instance, Staphylococcus-based food poisoning or botulism food poisoning, we call it foodborne intoxication because the majority of the effect is due to toxins. Now, how do we fight exotoxins? Our bodies will detect a toxin, which is a protein, as a foreign thing that shouldn't be there, and we'll make antibodies against it. So those antibodies are called antitoxins, and they provide long-lasting immunity. If we want to generate an artificial immunity to a toxin, then we would just take that toxin, we would extract it in some way, and often you can just do a heat step, or you can treat it with chemicals and slightly inactivate it, making a toxoid that doesn't have the same toxic effect, but still looks enough like the toxin to stimulate antibody production. And then you can inject that toxoid and use it as a vaccine. So this is what's been done for tetanus, when you have a tetanus shot, or the diphtheria shot that you get as a kid. Okay, so those are exotoxins. And remember, I just want to sort of stress this now, let me plant this in your head now. It's easy to inactivate them with heat. Exotoxins can be easily inactivated with heat. Now let's talk about endotoxins again. So here's the gram-negative cell wall. Here's our plasma membrane, the rest of the cells down here. Here's our little thin layer of peptidoglycan, our extra lipid bilayer, and then in the outer membrane, we have lipid A right here. That's going to cause us a problem. And so because this doesn't have an effect unless it's being released by a dying cell, the worst symptoms will happen when the cells are dying, either because our immune system's fighting them or because we're hastening their death using some kind of antibiotic. And this is why if you're treating somebody with a gram-negative bacterial infection, antibiotics can temporarily make that situation worse. Why endotoxins have this effect is that they stimulate macrophages, which we haven't talked about yet, but they're basically a type of white blood cell that is important. It roams through your body looking for stuff that shouldn't be there and engulfs it. Stimulates those macrophages to release high levels of cytokines, which I just defined like 2 seconds ago as these chemicals released by the immune cells, right? They're like chemical messages that the immune cell sends out when it sees something's wrong. It sends out a signal that travels through the body because it's soluble and calls other white blood cells to the site. And so this is actually similar to what those superantigens do, which is they cause an immune response. And so you get similar symptoms: fever, nausea, vomiting, diarrhea, sometimes shock and death. But you need a lot more endotoxin to have the same effect as a superantigen type of exotoxin. So one of the things that endotoxins can do is trigger fever, and this is just showing you how that works. Basically, here's the macrophage. It recognized this bacteria as being foreign, took it up, and chopped it up, as it's supposed to do. It takes it up and it digests it in the lysosome. The chopping up released the endotoxin, which caused the production of cytokines, an excess production of cytokines, that then travel throughout your bloodstream all the way to your brain and cause your body to increase the temperature. Now there is a way to detect endotoxins. and that is called the LAL assay. It stands for Limulus amoebocyte lysate. You don't really have to remember all that. That's kind of a mouthful, but you should know what the LAL is in principle, how it works. Basically, if you take someone's blood that has a lot of endotoxin, they have gotten sick or whatever, and there's endotoxin, and you mix it with the blood from a soft shoe crab, for instance, it will cause the blood to coagulate. And the reason is that in those kinds of crustaceans, their blood is different from ours. The blood cells are called amoebocytes. So this is not an amoeba. The blood cells are called amoebocytes because they can crawl. And for whatever reason, they make chemicals that interact with endotoxins. So there's historical reasons why people figured this out. I actually don't know what they are, but I can imagine all sorts of weird things like, I don't know, sick person on the dock, crabbing, cut his finger, notice something weird. But in any case, it's still used. If you extract this type of blood cells from these crustaceans and mix them with something that you worry is infected, you can find out if there's endotoxins there. Because what it does is it causes the blood to clot, or the amoebocytes, causes the amoebocytes to lyse and clot. Now, something that I want to mention is that endotoxins, the reason that people do this is because endotoxins survive autoclaving. So that means that if you are trying to sterilize something by putting it in the autoclave, you can kill the bacteria, right? But that isn't going to kill endotoxins because they're still present. Right? It kills the bacteria. The bacteria is not functional anymore, but the component of the bacteria that could cause you a problem could still be there. So you would take whatever material you're trying to test and put it through this assay to make sure there's nothing. Now, I want to emphasize this because you do have to know this. Endotoxins are heat resistant. Exotoxins are heat sensitive. So that is an important distinction. Exotoxins are much more likely to kill you, but they're much easier to get rid of. So this is just a chart that summarizes the differences, and I'm not going to go through this because we've talked about it, but you can have it in your notes as a way to study for these two types of toxins. Just to sort of walk through it, though, in brief, here is your typical exotoxin made by some sort of cell secreting exotoxin. Does anyone know what type of gram status organisms that make exotoxins typically have? Gram positive, that's right. And what is the relationship of the toxin to the microbe? It's made by the cell while it's still alive and secreted outside the cell. What kind of molecule is being secreted? What is this, actually? Is it sugar? Is it nucleic acid? What is it? It's a protein encoded by DNA. Now, if you can make an antitoxin, will it neutralize this type of toxin? The answer is yes. Typically, when you're dealing with a protein, it's pretty easy to make an antitoxin. Proteins just have these very distinctive shapes that our antibodies can easily recognize. Now, let's remind ourselves about this term. Remember LD50 and ID50? What was ID50 and what was LD50? ID50 was infectious dose, right? So meaning what? How much would you need to make some sample population, half of them sick, okay? That's the amount of the pathogen. LD50 always refers to toxins and stands for lethal dose. So what does that mean, LD50? How much do you need to kill half the population? And so if we're talking about exotoxins, do we need, relatively speaking, a little or a lot? Very little, yeah. So most of the organisms that make exotoxins are gram-positive. Not all of them, because remember that toxins are encoded by genes, which could be on plasmids or could be transmitted by phage. So they could certainly be taken up by a gram-negative. They're made by the cell. They're made of proteins. They can be neutralized by antitoxin. They have a very small LD50. What else do we know? If we wanted to inactivate them, what could we do? Remember that they're proteins. What makes proteins vulnerable? Heat. We could heat them. Or treat them with certain kinds of chemicals like bleach, for instance, which changes the pH, which also would cause a protein to fall apart. Now, endotoxins, on the other hand, are primarily associated with what? Gram-negative. In fact, exclusively, because it's a structural component of gram-negatives that you would not find in a gram-positive. Now, what is the relation of the endotoxin to the microbe? It's in the outer part of the cell wall. What kind of molecule is it? It's a lipid. Now, lipids are much harder for our immune system to detect. It's much harder to make an antitoxin, so they're not neutralized by antitoxins. Now, relative to exotoxins, is their LD50 higher or lower? It's much higher. Okay. So, where do bacteria get their toxin genes? That is, when they're making exotoxins, where are those exotoxins coming from? What kinds of, what is this term referring to? When you're sharing your DNA with your friends and relatives. So you could get it by transformation, right? By taking up a plasmid because the guy next to you died. You can get it by transduction, which means what? You got infected by a virus that happened to have just visited somebody who had that sequence and it got packaged into the viral capsid. You could get it by conjugation. So there are many plasmids that contain toxin genes. Tetanus neurotoxin is encoded on a plasmid. The Staphylococcus enterotoxin that causes Staphylococcus foodborne intoxication. Streptococcus mutans that makes a toxin that causes tooth decay has a plasmid that contains this gene. Some of the pathogenic strains of Staphylococcus aureus that cause flesh-eating bacterial disease have exotoxins that are on plasmids, and the enteropathogenic E. coli also has this on a plasmid. So we've talked about, I'm not going to go through this again. This is just to remind you that how transduction works, right? So viral infection, blah, blah, blah. This is how E. coli 0157 got its Shiga toxin. That's how Vibrio cholera that causes cholera got its enterotoxin. And that's how botulism got its neurotoxin. As well as bacterial pneumonia, its virulence factor, this capsule, is also acquired by a viral infection at one time. Diphtheria toxin is another example. So this is just showing you some of the organisms or some of the pathogens that receive toxins because of a viral infection. So diphtheria, Streptococcus pyogenes, Clostridium botulinum, Clostridium tetani, Vibrio cholerae, and Staphylococcus aureus, and talks about the different kinds of exotoxins that they have. Now, that's all about bacteria, basically. Viruses will cause the symptoms that they cause in a different way. So the first thing that's different is that they have to get into the cell, and as our polio people just reminded us, they get into the cell by having attachment sites on the surface of the virus that bind to cell receptors and trick the cell into taking the virus into them. What happens next is usually the result of the virus multiplying inside the cell and making proteins that have various effects in the cell. And this suite of effects is called cytopathic effects, so effects that are happening in the cytoplasm. So that includes the production of virus particles, which in and of itself, it's like you're completely taking over whatever function this cell had and turning it into a virus-making factory. And you can see it actually, because there will be these regions where these viral particles are highly concentrated called inclusion bodies. Because the virus is taking over production for itself, it prevents the cell from doing any of its normal DNA, RNA, or protein synthesis. And in order to get out, the virus often will explode the cell, which isn't so great for the cell. So the cytopathic effects are caused by lysosomes that release enzymes. So as the cell is dying, it's releasing its contents, which are digestive enzymes, basically. It often causes cells that are next to each other to fuse together into what's called a syncytium. Now, why would that be helpful for a virus? Why would it have evolved the ability to cause cells that are next to each other to just fuse together in one big compartment? How does that help viruses? Now they have, it's like a territorial takeover. You know, they're in this one cell. By causing the cells to fuse together, they just gain all of that territory and all the resources all of a sudden. It obviously changes the host cell function. One thing that does happen is that cells that are infected with viruses will produce interferon. We'll talk about this again when we come to chapter 16, but it's kind of the cell's cry for help. It can often cause chromosomal damage because the DNA isn't being properly taken care of anymore. It will start to fall apart. The other thing you'll notice is loss of contact inhibition. And I don't know if you remember this picture that I showed you in one of the previous lectures, I think in chapter 13. I showed you a picture of a cell culture and next to it a virally infected cell culture. And the virally infected one, all the cells were kind of rounded up. And so they have different kinds of relationships to each other. And one of the things that's changed when you have a viral infection is something called loss of contact inhibition, which just means that the cells don't behave normally with respect to their neighbors anymore. And I'm not going to go into that in more detail, but it's actually pretty cool. Okay, so the last thing we're going to talk about before we move on to the lab are the final sort of suite of effects that are caused by pathogens that are not bacteria and not viruses. So that would include things like what? What's left that can make us sick? Obviously fungi, yes?
The virus and the bacteria. So usually when we get the bacteria, it usually gets fever, right? But when we get the virus, it usually gets fever.
Well, there are viruses that will cause fevers. Like flu, if you've had the flu, that causes a pretty high fever sometimes. So the fever, I mean, yes, there's certain kinds of bacterial pathogens that will cause fever, either because of an exo or endotoxin. So if it's an exotoxin, that's a superantigen, it will cause like fever-like symptoms. If the endotoxins will cause fever. Viruses cause fever in a different way, but basically it's because the virus causes an immune response, and the immune response is actually producing the fever to try to kill the pathogen. So the fever is something our bodies will actually do to try to, it's almost like a homegrown chemotherapeutic. We raise the temperature enough to try to kill whatever's happening before it kills us. Right? If you overshoot, too high, the fever can kill you or cause brain damage. So many pathogens will cause fever, but both viruses and bacteria, depending. So we've got fungi that could be pathogens as well, and also a few others. You'll remember from your Kingdom of Life in the very beginning, I sort of said these are all the different topics we're going to cover, so there's a few we haven't covered yet. So we'll start with fungi. Now, fungi many fungi don't, but some of them are pathogenic. But usually they don't have toxins. There's a few exceptions, but most of them don't have toxins. Most of the time, the problem with the fungus is that by growing in whatever part of your body, they're producing a lot of waste, and the waste is what causes damage. Also, the fungus can cause us to have a chronic allergic response because it's detected as foreign. There are also some enzymes that are made by fungi, such as proteases, which by the name you might guess actually chop up proteins. That's not always very good for you. And candida, if you've heard of candida, it's a yeast which can cause systemic infection, and it has proteases that are not good for you. Also, there can be fungi that are protected from phagocytosis, which is how our body normally would try to clear cells that shouldn't be there. Now, as I said, some fungi do encode toxins, so here are the exceptions. Ergot toxin, causing hallucinations, is made by this fungus. And you may have actually heard of this. Aflatoxin in peanut butter is made by Aspergillus. So sometimes you can have toxins in peanut butter. And mycotoxins, which literally means toxins made by mushrooms. There are some mushrooms we know that are deadly, right? You eat the wrong mushroom, you can die. And that's because, for instance, this mushroom phalloidin, there are mushrooms that make phalloidin or amanitin, both of which are neurotoxins. And they're made by this species Amanita phalloides. Now, also there are pathogens that are protozoans, which means what? What kind of cell are we dealing with? What kind of, how would we compare protozoans to, say, bacteria? They're eukaryotes. How would we compare them to us? They're single-celled, okay? So again, like the fungus, largely the problem is the result of waste products that are made by the protozoans that cause symptoms. And a lot of them will avoid the host defenses either by growing inside of white blood cells or by changing their outsides. So there are things on the outside of these protozoans that can be detected by the immune system when we'll make antibodies, and then they'll just be like, okay, well now I'm going to change that thing, now I'm invisible.
Just like before this, there was one thing that makes oxygenation. Is that the one that people use as a drug?
Yes.
That's the one?
Yes, it is. Yeah. All right. Now, examples of protozoans, Plasmodium is one. What disease does that one cause? Anyone know? It's on your disease list. Malaria, exactly. So here's our hint is that it's transmitted by mosquitoes, although obviously we know many things are transmitted by mosquitoes. And Plasmodium reproduces in the host, and that causes an illness. Giardia, some of you maybe if you go camping, you'll be aware that you shouldn't drink untreated water because of this organism, Giardia lambulae, which is a single-celled eukaryote, looks like this. And basically, the symptom is diarrhea, and it digests both the cells and tissue fluids, and so causes damage directly and also indirectly. Sleeping sickness caused by trypanosome, so African trypanosomiasis, as well as in the US, a version of that is Chagas disease, also reproduces inside of host cells. This is the adult form of one of the insect vectors, and this is what the trypanosome, the single-celled protozoan, looks like. It looks like a little fluke almost. But it reproduces inside of the tsetse fly, and then if you get bitten, you get the disease. Its main method of evading detection, and this is just a schematic that shows the surface of the trypanosome in various stages of the infection in one host. So initially it comes in and it's got, various things on the surface that can be detected as antigens by the immune system to make antibodies, and you start to clear it. But in the meantime, it's been mutating, and one of the mutant versions begins to escape being detected, and then it gets really high, and then we find it again, and we make antibodies against it, start to kill it, but meanwhile it's been reproducing and mutating, and so that guy is, and so on, it goes like that. So it's a difficult one to get rid of. We also have some of our pathogens are worms, basically, and they're included in microbiology because the early stages of the worm life cycle is microscopic, even though the adult is a eukaryote with multiple cells. And typically their effects are just that they're using up our resources, and they may cause some tissue damage. Their waste products can cause symptoms that are uncomfortable or cause malnutrition because they're using up the food that we're trying to consume. And this is just because, in fact, tapeworms have been used as a diet tool. There are several different kinds of tapeworms, and there's one that causes the least toxic effects, and so that's one that has been actually sold for weight loss because you can relatively easily, you can ingest it, you can lose weight, and then you can relatively easily clear it with drugs. Not recommending that by any means. It's disgusting, but it has been used in that way. There's also some algae that produce toxins, and so you may have heard of red tide, right? That's the result of algae that makes these the saxitoxin. Okay, last thing we need to talk about how the pathogen gets out. Why do we care about this? from a public health perspective, why do we care about how the pathogen's getting out? Because it's how it's transmitted, right? So, for instance, if it's something that is in the respiratory tract, then most likely it's being transmitted through coughing and sneezing. If it's in our GI tract, it can get into the saliva or feces, and then we have this oral fecal transmission route. Genitourinary tract infections are typically STDs, are sometimes transmitted in the urine. And there's skin infections, right, that can be transmitted by direct contact between human skin. There are also blood vectors that are important, so either things that are transmitted by insects or by puncture, and by misusing contaminated needles and syringes. All right, so that's all we need to talk about. We're going to take a 5-minute break, and then we are going to get started on